Okay, guys. Well, as you can see, we have no projector again today, so they're on their way to deal with that, which means I get to wing it. Yeah, I don't like it any more than you. I don't like it any more than you do. So, reminder, of course, that tomorrow is our exam, and the exam is written and done and so forth. I think it's pretty straightforward. I did make more short answer questions and fewer long answer questions. So there's 20 short answer questions on this exam, which is more than we had last time. And there's only 2 longer answer questions for the exam. Okay. Let's see. What else can I tell you? The answer to it? Well, then everybody would get 100. Then there is an extra credit question on there. I'm sorry. I thought you said the answer to it. I was going to say that wouldn't help. Everybody gets 100, then nobody's grade changes, right? That'd be kind of boring. What's that? You want a word bank? Well, I've already given you all those, right? Just go back and take every word that I gave you, and you have a word bank, right? Okay, so I don't mean to be snarky, but anyway, I think it's a straightforward exam. So I know you guys are working on it hard, and I hope that you will do well. I really do. Yesterday I finished talking about gluconeogenesis, which is where the material for the exam stops, and I got the video and the review session and the notes posted late this morning. So if you haven't had a chance to look at all those, they are there and available. There was one thing I didn't mention that I'll mention here, and I won't make it on the exam, and that had to do with the fact that Pyruvate Kinase is the 3rd regulated enzyme there, and I mentioned it briefly yesterday, but I didn't really say too much about it. So I'll say a little bit about it here. One is that Pyruvate Kinase, as I noted, is the last enzyme in the glycolysis cycle. And it's unusual for a cycle to regulate its last enzyme, but that last step of glycolysis is also an unusual reaction. That's the Big Bang. And the problem for a cell is that the cell is having to bypass that reaction in order to make glucose. So if you recall, the cell is having to go from pyruvate to oxaloacetate to PEP in the process of gluconeogenesis. And that took the enzymes pyruvate carboxylase and PEPCK, phosphoenolpyruvate carboxykinase. Now, the end product of those 2 reactions is PEP. And so you can imagine that if PEP is present and pyruvate kinase is active, what do you suppose is going to happen? It's going to go right back to pyruvate. You will have burned 2 triphosphates to make PEP. You would gain 1 triphosphate going back to pyruvate. So you would be losing energy. You would be generating heat. And you would get absolutely nothing if pyruvate kinase were active. So the reason the pyruvate kinase gets regulated is because you have to turn it off in order to do gluconeogenesis. Now, what I just described to you is a phenomenon that is a very important thing in control of metabolic pathways, and that's something called a futile cycle, F-U-T-I-L-E. A futile cycle occurs when catabolic and anabolic pathways are going on simultaneously in the same cell. A futile pathway occurs when catabolic pathways are going on simultaneously in the same cell. They always produce the same thing: no net products, loss of energy, and heat. Okay? Well, it turns out that that actually, in some cases, is beneficial. Some organisms actually run futile cycles to generate heat. Organisms that are out in very cold environments sometimes will run a futile cycle so that they produce heat that's necessary to keep the body warm. Okay? So that's one thing. But usually cells don't want to run futile cycles because they're not productive for any reason. Another thing I didn't mention yesterday about gluconeogenesis that I'll mention here is the fact that gluconeogenesis takes 6 ATPs to make each glucose. 6 ATPs to make each glucose. It also requires 2 molecules of NADH. So what we see from the synthesis of glucose via gluconeogenesis is that it takes more energy to make glucose than the body gets out of oxidizing glucose. And that sort of makes sense. It's why we have to continue eating. if we could get as much energy out of glucose as we put, much energy out of glucose as we put into it, then we would never have to eat. We would have this continual cycle of all these things going on. But in fact, we have to eat because we lose energy every time we try to make glucose. Okay, so that's one important consideration. Another pathway that I want to talk about, that's what I want to say basically about gluconeogenesis. As I said, this last portion I won't hold you responsible for on the third exam. Excuse me. The next pathway I want to talk about is one that's actually one of the most complicated pathways that we have to talk about, but I'm going to do it in very simple fashion. It's called the pentose phosphate pathway, PPP. Okay? The pentose phosphate pathway, as its name suggests, is a pathway that involves metabolism of 5 carbons. Okay? Now, it's actually quite a bit more involved than that. I should say not just 5 carbons, but sugars having 5 carbons. Okay? It's actually quite a bit more involved than that because when we look at the pentose phosphate pathway, we actually see 6 carbon sugars. We see fructose and we see glucose present and being oxidized. We see 5 carbon sugars. We see 4 carbon sugars. We see, 3 carbon sugars. And we even see a 7 carbon sugar. It's one of the few places where a 7 carbon sugar actually appears in metabolism. Okay? So the pentose phosphate pathway is called the pentose phosphate pathway because one of its very important products is a pentose. Pentose being a 5 carbon sugar. And one of those 5 carbon sugars that's produced by the pathway is ribose. So ribose is produced by the pathway in the form of ribose 5-phosphate. You can call that R5P. Hi, thanks for coming over. R5P. And ribose 5-phosphate, of course, is necessary for the synthesis of nucleotides. Necessary for the synthesis of nucleotides. Now, the pentose phosphate pathway, however, is of other importance to the cell. Now, the pathway is unusual in the sense that it doesn't have a beginning and it doesn't have an ending. Whoa, right? I like to think of the pathway as sort of an intersection. Okay? If we have, let's say, we're traveling to Portland and we're going over the bridge at I-5 and we see all the different things going various different directions. Depending upon which exit you get onto, you can go north, you can go east, you can go west, you can even turn around and go south if you pick the right exit. The pentose phosphate pathway is like that. It can take inputs from any of these sugars that I talked about. So a 6 carbon sugar can be a starting material. Oh, thank you. So what did you do?
Apparently someone was using a laptop connection.
Oh.
This. Yeah, apparently we're doing the same thing with the audio. So if this is ever an issue, I would look here first, make sure none of these connections are connected to the computer, and make sure that this is.
And I want the audio. So is the audio here?
Yes.
Okay, great. Okay. All right, thank you very much. Appreciate it. Yeah, take care. Okay. So the pentose phosphate pathway. Now, I said it doesn't have a beginning and it doesn't have an end. This is, I'm sorry? A little bigger? Not much, but I can make it a little bigger. Hopefully you've got the image from my notes. Okay. Where'd my pointer go? There's my pointer. So let's talk then about the inputs. There's glucose 6-phosphate. It's a 6 carbon sugar. There's 6-phosphogluconate. Now I'll talk about that in a second. That's an oxidized form of glucose 6-phosphate. All right. Ribulose 5-phosphate. That's a 5 carbon sugar. Ribose 5-phosphate. There's a 5 carbon sugar. You're not going to have to know these names of all these things. Okay. So I'm not asking you to do that. I'm just pointing them out to you. Here's a 7 carbon sugar. It's called acetoheptulose 7-phosphate. You don't need to know that. Erythrose 4-phosphate, 4 carbon sugar. Xylulose 5-phosphate, another 5 carbon sugar. So anyway, all these various sugars are here, okay? And any of them can be something that starts, if we want to think of starting the pathway. So let's say the cell has just ingested a giant molecule of DNA. or an RNA. All right? And it has no need for that DNA because it has its own DNA. So what's it going to do? It's going to take those nucleotides and it's going to break them down. And when it starts breaking them down and it breaks them all the way down, you're going to get the sugar that's in them, ribose 5-phosphate. Well, because of the pentose phosphate pathway, the cell can take that ribose 5-phosphate and it can use it in this cycle to make anything else. It can use ribose 5-phosphate to make glyceraldehyde 3-phosphate. It can use it to make fructose 6-phosphate. It can do any of these things with it. So this is one starting point. If the cell had a lot of erythrose 4-phosphate, again, comes in here. Or what if the cell needs erythrose 4-phosphate? Erythrose 4-phosphate is actually used for making some of the amino acids. It's used to make tryptophan. Well, if cell needs erythrose 4-phosphate and it's got a source of ribose 5-phosphate, it could use this and make this. Input, output. So there's no fixed input to this pathway. There's no fixed output to the pathway. The inputs rely on what the cell has an abundance of, and the outputs depend upon what the cell needs. That's pretty cool. Okay? So if there's one message I want you to take away about this pathway, that's it. Inputs depend on what the cell has an abundance of, and the outputs are what the cell has need for. Now, there's a couple things I want to show you here that's kind of cool. So one of the main products of the pathway I told you is ribose 5-phosphate. Cells tend to need a fair amount of ribose 5-phosphate because they need to make nucleotides, especially if they're planning to divide. So ribose 5-phosphate is a pretty constant need that cells have. That means it's a very common output of this pathway. That's why it's the pentose phosphate pathway. Another thing that's produced by this pathway in abundance is NADPH. NADPH. Now, NADPH, we will see, is needed for making fatty acids, among other things. Cells don't use NADH to make fatty acids. They use NADPH. Okay? Well, this is a prime source of that, and that comes from a couple of oxidation reactions. One oxidation reaction to oxidize glucose 6-phosphate, and a second one to oxidize its product. So that's the two oxidation reactions that can produce NADPH. So NADPH is the second output of this pathway. Okay? The third output of this pathway is anything that's there, as I said. So outputs depend upon cell needs, and because of this, cells can make a variety of things as a consequence of just that. Okay, questions about that? We will see when we talk about photosynthesis, for those of you who are botany plant pathology, that the Calvin cycle, which is also known as the dark cycle of photosynthesis, has enzymes very similar to this, and in fact has many reactions similar to the pentose phosphate pathway. Okay. Let's go back here. One of the things, actually I should show you this. This is fine here. So if we simplify and summarize what's happening in the pathway, we see that what happens is that in the cell, the pathway's ability to shuffle between sugars that have 4 carbons and 3 carbons and 7 carbons and so forth, is their ability to move 2 carbons at a time, or in some cases 3 carbons at a time. Okay, so here's let's see. Here's a good example of a 2-carbon shift. Okay, I've got 2 5-carbon molecules. I take 2 5-carbon molecules and I pop off the top 2. I use something called a transketolase to do that. I put 2 of those onto the 5, I get a 7, and I get a 3. Okay? So transketolase and transaldolase reactions are very helpful for cells to being able to do things as they need. Here's a 3-carbon shift. In this case, 3 carbons of this guy are taken and put onto here to make a 6-carbon molecule, and that leaves behind a 4. Right? So this is a product of a transaldolase reaction. Now, I'm not going to ask you to draw this table, but I think you should know that transketolase and transaldolase allows the pentose phosphate pathway to shuffle carbons. And that will become important when we talk about the Calvin cycle in photosynthesis as well. Okay? So transketolase and transaldolase allow the cell to do shuffling of carbons from one molecule to another. Now, we're actually, despite starting late, we're actually going to get done early today so we can get out and enjoy the weather. And I figure you guys will want to have a little bit more time to study anyway, so we're not going to, this lecture won't take too long. All right, when I talked about the structure of carbohydrates, I said one of the polysaccharides that was important for us to know was glycogen. And glycogen, you recall, was a molecule that was a storage form of glucose in animal cells. And the reason it was important for animals was because it had all these branches and as a consequence had all of these ends. Every blue guy there is an end of a glycogen molecule. And I told you that it was important for animals that they have a storage form that has as many ends as possible because animals get glucose out of here by breaking down this molecule starting at ends. So the more ends you have, the more things that you can start to break down. So that's what we're going to focus on now, is how it is that the body breaks down and makes glycogen, okay, for its various purposes. Okay. Well, glycogen metabolism, the glycogen metabolism pathway, fortunately, is one of the easiest pathways you'll ever learn, okay? Because there's only just a couple of steps in the whole thing, both the synthesis and the breakdown. Here is a step in the process of making, okay, an intermediate in glycogen metabolism. Okay, actually, I'm sorry, this is the breakdown. So this is a step that they're very similar. If we reverse it, essentially we have the other one. But let's look at the breakdown of glycogen. So here's a glycogen molecule that has all these glucoses in a polymeric form in glycogen. Okay? If we take a molecule of phosphate, okay, and we mix this mixture with an enzyme called glycogen phosphorylase. Some of you have asked me, what's a phosphorylase? Okay? A phosphorylase is an enzyme that uses phosphate to break something. A phosphorylase is an enzyme that uses a phosphate to break something. The phosphorylase is using this phosphate to break off a molecule of glucose 1-phosphate. That's what glycogen phosphorylase is catalyzing. What's left behind is a glycogen that has one fewer glucose in it. Okay? Now, this can readily be made into glucose 6-phosphate, and glucose 6-phosphate is readily used by what pathway? Glycolysis. Okay? Bang, bang, we're done with the pathway. There's one other consideration in breakdown that I'll show you, but it's not complicated. Okay? One other consideration. But you can see this is very simple. There's also an advantage to this. The advantage. Look how this cell is using phosphate. You don't see any ATP here, right? When I went from glucose to glucose 6-phosphate, I said that took ATP to put that phosphate on there, right? Here, I see a few heads nodding. Yes, it required ATP to put that phosphate on there. Yet here I'm putting a phosphate on without any ATP. Did I lie to you? How am I able to put a phosphate on here where I wasn't able to put a phosphate onto glucose? Any thoughts? There is a lower G0 prime, but why? The glycosidic linkages, okay, he's exactly right. The glycosidic linkages between the individual glucoses contain energy. And that energy is used to drive this reaction. That's why this happens. Now, this is really cool because it means that the cell, which is really needing energy, isn't having to use ATP to put a phosphate on, right? This gets converted to glucose 6-phosphate, and we're already off and running in glycolysis without using any ATP. So this is a very efficient way for the cell to manage its energy. Now, I said there was one other enzyme we had to think about, and by the way, here's the reaction: glucose 1-phosphate to glucose 6-phosphate. Okay, and that is a phosphoglucomutase that catalyzes that. Does that tell you anything about what the structure of the intermediate might be? The intermediate is a glucose 1,6-bisphosphate, right? So it puts both phosphates on, then it pulls this one off, and it's left with this one. So whenever you hear the word mutase, that should tell you something about how it works. Okay. The only other consideration in glycogen breakdown is the fact that glycogen phosphorylase is a finicky enzyme. It's a finicky enzyme. Here's a molecule of glycogen. Remember, glycogen has branches. So here's one end, and here's a free carbon number 4. Alpha 1,4, alpha 1,4, alpha 1,4 linkages all the way through here. Okay? Here's a branch, an alpha 1,6. Okay? So a glycogen molecule has all kinds of branches. However, glycogen phosphorylase is finicky. It won't cut within 4 glucoses of a branch. It won't touch it. Okay? It's like when you're little and you don't want to eat your peas, right? When I was little, I didn't like to eat peas. As an adult, I eat peas. You think I'm crazy, but it's true. I eat peas with almost every dinner. I love peas, right? I'm not a finicky eater as an adult, but I was a very finicky eater as a kid. Well, this enzyme is a finicky enzyme, and it doesn't change over the life of a cell. It ends up being finicky all the time, right? Okay, so because of that, if the cell didn't have a way of retrieving those other glucoses there, glycogen wouldn't be very useful. Well, fortunately, the cell has a way. It has an enzyme that will actually it does 2 crazy things. Instead of just cutting this thing off here, it does the following thing. It cuts cuts off the terminal 3, and transfers them down to the end of the other chain. That's kind of odd. And then it takes this last one and it cuts it off using water instead of using phosphate. Now, I would ask you why it does that, but since I think all of you are already thinking about stuff for the exam, I won't press you here. But there's a reason why this enzyme, by the way, this enzyme is called debranching enzyme. Glycogen debranching enzyme or just debranching enzyme is fine. You can call it debranching enzyme. Okay? There's a reason why the cell does this, or why the enzyme does this. Remember that there's energy in those alpha 1,4 bonds. If the enzyme were to cut it here at the 1,6 position and move it down, it wouldn't have sufficient energy to make a 1,4 bond down here. That is to say, an alpha 1,4 bond has more energy than an alpha 1,6 does. So it's not practical to take a lower energy bond and use it to drive the formation of the higher energy one here. It wouldn't be practical. Now, there's actually evidence that that's the case from what I've already told you. Because the enzyme uses water. It doesn't use phosphate. There's not enough energy in this alpha 1,6 bond to put a phosphate onto that guy. That's why it uses water, and that's why the product of this is glucose, not glucose 1-phosphate. Make sense? Everybody's just kind of staring at it, like you've been staring at your notes for some time, right? All right, so that's what glycogen debranching enzyme does. That's the total of glycogen breakdown right there. Let's talk about synthesis. It's almost as simple. It's got one added step. The one added step is this molecule right here. It turns out I lied earlier. Glycogen synthesis does not have glucose 1-phosphate being the adding molecule to put the glucose onto the glycogen. it uses this molecule instead. We'll talk about it in a second. Okay? This molecule is an example of what I call an activated intermediate. An activated intermediate. It's a molecule that has a high energy bond, and it uses the energy of that bond to transfer a part of itself onto something else. What do you suppose it's transferring? Well, looky there. There's glucose. This molecule transfers glucose onto a growing glycogen chain. So UDP glucose is needed as a substrate to put glucose onto a growing glycogen chain. This high energy bond drives the formation of the alpha-1,4 linkage in glycogen. It drives the formation of the energy-rich alpha-1,4 linkage in glycogen. What's the enzyme that catalyzes that reaction? The enzyme that catalyzes it has a very simple name. It's known as Glycogen Synthase, Synthase. Glycogen synthase uses this as a substrate to add glucose to a growing glycogen chain. The product of that guy is UDP. That's what's left. Yes, Mariah. Well, you're getting ahead of me here. The debranching enzyme is for breaking down glycogen. So we have to make branches, and we make branches with something called branching enzyme. Okay, but yes. This happens before branching enzyme gets there. That's right. John?
To be clear, the glycogen synthase uses this molecule to add glucose to the chain.
That's correct. Uses this molecule to add glucose to the growing glycogen chain. Okay, here's the reaction right here. All right. Here's our UDP glucose. Here's the end of a glycogen chain. Here's the addition of the molecule right here. And we've seen now it's grown by one additional residue. All right. So that's what's happening in the glycogen synthase reaction. And no, you don't need to know there's an oxonium ion intermediate or any of that stuff. Well, Mariah had the right idea here, because if all I did was make alpha-1,4 bonds, I wouldn't have any branches. Wouldn't have any branches. So there must be some enzyme that makes branches to produce the final glycogen. And there is. There's an enzyme called branching enzyme. Branching enzyme. Branching enzyme. By the way, books get all hot and bothered by how many from the end it cuts in and where it puts it and so forth. It makes branches, right? We're not going to worry about that. It makes branches, okay? So branching enzyme makes branches. What does it do? It breaks alpha-1,4 and makes alpha-1,6. But we already know that 1,4 bonds have more energy than 1,6 bonds, so we know that's a practical reaction for the enzyme to catalyze. No, alpha-1,4 bonds have greater energy than 1,6. So that means it's practical to go from a 1,4 to a 1,6. I think I said that right, right? Okay. So if we just compare the energy of an alpha-1,4 bond to the energy of a alpha-1,6 bond, the alpha-1,4 bond has more energy in it. We had to use the UDP glucose to make that bond, okay? So if this has more energy than a 1,6 does, it means that the energy released from this can be used to make this. And since this is greater, then that is a favorable reaction. The benefit of the cell is that it's making branches. Right, because the more ends that it has, right, the better off the cell is. Yep. Now this reaction goes on primarily in liver and muscle cells. Muscles make glycogen. They can't make glucose, but they make and they store glycogen, because muscle cells need a lot of glucose, and when the time comes for them to contract, they need to break down glycogen quickly to release glucose so that the cell can do glycolysis. Okay. Now, the last thing I'm going to talk about today is this rather confusing figure that I showed you earlier in the term. I'm going to spend just a few minutes talking about it, and then I'm just going to let you go early. Okay? So, this figure shows the regulation, okay, of a molecule known as of the enzyme known as glycogen phosphorylase. Now, I call it glycogen phosphorylase. Many people call it simply phosphorylase. Right? But let's take some time and understand what's going on with this molecule, or this enzyme, I'm sorry. So remember, what's glycogen phosphorylase? Glycogen phosphorylase catalyzes the phosphorylasis of glycogen. That is, it uses a phosphate to break off a glucose 1-phosphate. That was the first step in the breakdown that I showed you earlier. Turns out this enzyme is regulated in a fairly complicated way. It's one of the most complicated regulations we're going to see, and we're not going to go into all the depth of the regulation. But there are some highlights here that I want you to understand. Okay? All right. 1, the enzyme can exist in a T state and an R state. T state, I hope you remember, is the less active form. The R state, the more active form. The enzyme can also be covalently modified. It can have no phosphate, in which case we call it phosphorylase B, or it can have 2 phosphates, in which case we call it phosphorylase A. Now, both phosphorylase A and phosphorylase B can exist in the R state and the T state. And in both cases, the T state is the less active form than the R state is. Everybody with me so far? Yes, question.
How many phosphates does the phosphate or the phosphorylase A have?
Phosphorylase A has 2 phosphates. B has no phosphates. So the first question is, well, how do the phosphates get there? Well, I hope in the back of your mind you remember what a kinase is and what a kinase does. You know it's got to be a kinase to put a phosphate on, right? The kinase that puts that on is called phosphorylase kinase. It's very simple. Phosphorylase kinase puts a phosphate onto phosphorylase. What takes the phosphate off? Well, I hope you remember again that a phosphatase is something that takes a phosphate off. In this case, the enzyme is called phosphoprotein phosphatase. How about I let you call it PP1? Okay? PP1. Everybody with me now? Now, I think, okay, I'm going to talk you through something here, and I'm going to repeat it in the lecture on Friday, because this is complicated. Okay? But I'm going to say it here to introduce the topic to you and let you think about it. Okay? You'll notice that over here we see glucose. Over here we see ATP and or G6P, and you see AMP. What are those? What does it take to go between R and T state for any enzyme? It takes allosteric effectors. Allosteric effectors bind to an enzyme and affect its activity. And they do that by converting an enzyme between the T and the R state. Notice that T and R state does not require phosphorylation. Phosphorylation involves other things. So to go from T to R, I need allosteric effectors. All right. Well, what affects phosphorylase B? Phosphorylase B is what people tend to refer to as the less active form of glycogen phosphorylase. The less active form. Why do they call it the less active form? Well, we'll see why in a second. What converts the T form into the R form? Well, that's AMP. does AMP indicate in the cell? Low energy. Cells rarely have much AMP. That means that usually this guy doesn't go into the R state. Usually, phosphorylase B is found in the T state. Not always, but usually in the T state. When might it be in the R state? Well, let's say I take off and I go sprinting out of here because the building's on fire. I got to get out of here as quickly as I can, right? My muscles are, I've already said my muscles are going to burn ATP real quickly, okay? And when they start running out of ATP, they start burning ADP, and when they burn ADP, they make AMP. Now, if I didn't have alternative sources, remember I talked about creatine kinase? If I don't have alternative sources of energy, what's going to happen to those muscle cells? Creatine kinase is going to carry me for a little ways. However, I need glucose. When AMP in that scheme starts building up, what's my glycogen phosphorylase going to do? It's going to flip into the R state, and it's going to start breaking down glycogen. I'm therefore going to have glucose for glycolysis. It's not a last resort. It's actually a very important step. It's just that unless I'm doing what I just said, a lot of just heavy exertion, I'm not making much of this. But I want this there when I'm doing that heavy exertion. What about this? Well, these guys convert the R state into the T state. Cells generally have plenty of ATP. Every one of you sitting around in here right now has high levels of ATP because you're not burning it with your muscles. You're not, except writing with your hand or maybe doing a little thinking in the old brain, you're not burning a lot of ATP. Your ATP levels are high. Your glucose needs are low. So your glycogen breakdown, at least from this side, is stopped. G6P we won't talk about here, but it's also an indicator of high energy. High energy. Okay? So high energy things flip phosphorylase B into the T state. Low energy flips it into the R state. This is the cool one over here. Glycogen phosphorylase A is what people refer to as the more active form of glycogen phosphorylase because usually it's in the R state. It's not usually in the T state. Why is that? Well, the only time the enzyme gets flipped from R to T is if glucose concentrations in the cell are high. That doesn't happen very often. When cells get glucose inside of them, what do? They do glycolysis. Glucose is a poison, remember? Cells deal with that poison. They don't have high concentrations of glucose. So most of the time, there's no glucose. This doesn't go up. Now, you notice there's nothing over here that's an allosteric effector. What does that mean? It means that this guy, in the absence of glucose, automatically flips into the R state. So if there's no glucose present, glycogen phosphorylase is going to be present in the R state. Now, I'm going to repeat that next time. Yes, sir? When a person runs out of glycogen, where does the body go to next? So if you run out of glycogen, if you're completely out of glycogen, you're in pretty deep doo-doo. So you're in starvation mode. Your body would then go to using amino acids to make glucose. Okay? That's what they would do. Because we can't use fatty acids to make glucose. They would use fatty acids for basic energy, but fatty acids would not provide the source needed to make glucose. So you would start breaking down your protein to make glucose if that were happening. And that's what happens on a starvation diet. Yeah. Now, the last thing, I promise, this is the last thing, right? And I'm going to repeat this next time also. Okay? This guy right here. this process going to the right, this guy gets activated by adrenaline, epinephrine. The reason that the person who is on adrenaline can do incredible feats is because this enzyme becomes very active. It makes this, which makes this, which breaks down a lot of glycogen, makes a lot of glucose, and muscle cells are very happy. This guy gets activated by insulin. Notice they're doing opposite things. This guy is activated by insulin. Insulin says, hey, there's too much glucose in the bloodstream. Let's have our cells all suck it up so the blood glucose level fall. Great. However, when all that glucose gets inside of a cell, the cell says, glucose is a poison. Do I want to be breaking down my glycogen if I've got plenty of glucose? Absolutely not. And what's glucose going to do? Right here. Okay? Now, we'll talk about this. This is called hormonal regulation of pathways. We're going to talk about this next time. And I said I'd leave you out early. And what I'll do is we'll save the song till next time. How about that?